Okay, now we're going to talk about peptide hydrolysis and denaturation. Hydrolysis is the process in which we are breaking the peptide bonds in a protein by treatment with either aqueous acid or base, or as our bodies utilize certain enzymes. In the body, we have an enzyme called peptin in our gastric juice, and that is responsible for hydrolyzing some of those amide bonds in large proteins to make smaller, more manageable peptide chains that are then transported or moved through the intestines, where we have two other enzymes, trypsin and chymotrypsin, that are responsible for hydrolyzing the remainder of those peptide bonds, resulting in individual amino acids. This diagram or pictorial shows the structure of this tripeptide, isoleucine, gly, and phenylalanine, or glycine. And we have two peptide bonds holding these three amino acids together. And during protein hydrolysis, which of course is going to involve water, we're going to break that peptide bond and release individual amino acids. Denaturation, on the other hand, is the process of disrupting quaternary structure if we have a protein that has that type of structure, tertiary structure, and secondary structure. It does not, however, affect primary structure. So the denaturation is just disrupting those intramolecular or intermolecular, if we've got a protein that has quaternary structure, disrupting those interactions and opening or altering the shape of that protein. So we mentioned acid and base a moment ago, but high temperatures and even strong agitation can disrupt those non-covalent interactions. And again, those are going to be things like hydrogen bonding, electrostatic interactions, disulfide bonds, or well, they're non-covalent, so take that back. You do need another agent for that. But those non-covalent interactions that contribute to the shape of that protein. Once we have denatured a globular protein that is typically water soluble, because remember those polar amino acids are going to be on the surface and those nonpolar amino acids are buried on the interior, it will become less water soluble. Ovalbumin is a good example of a process of denaturation that I'm sure every one of you have seen. It's the major protein in egg whites. It makes up about 80% of egg white protein, and it is denatured when the egg is cooked. And so what we see is a change from that gelatinous, clear, colorless substance converting to an opaque and white substance during that denaturation process. So enzymes are utilized by living organisms to catalyze a number of biological reactions. Enzymes are proteins. They function to increase the rate of a reaction up to 10 to the 6 to 10 to the 12 times faster than that reaction would be without the enzyme. However, they are not themselves changed. So if you remember what the definition of a catalyst is, it is a molecule that increases the rate of the reaction, but is not consumed in that reaction. Enzymes are very specific. They catalyze a certain reaction or type of reaction only. And most enzyme names end with a suffix ase, like peptidase, lipase, hydrolase. So we can tell we're dealing with an enzyme if it ends with ase. Often cofactors are required by enzymes. Cofactors can be metal ions or organic molecules, and we'll look at both, or I think we'll see both of these examples, and they are required for that enzyme catalyzed reaction to occur. NAD is an example of a cofactor. Nicotinamide adenine dinucleotide is its big name. So we just typically shorten it to NAD, and it has a positive charge. It's a cofactor or coenzyme, and it oxidizes lactate to pyruvate. And the enzyme involved here is lactate dehydrogenase. So here we have a lactate molecule. Here we have our coenzyme, our enzyme, and it's these hydrogens that are going to be removed. Remember that oxidation is the loss of hydrogen and the gain of CO bonds, and that's exactly what happens here. So lactate is oxidized to pyruvate, and if something's oxidized, that means something else has to be reduced. So what gets reduced is our coenzyme, and NAD plus then is reduced to form NADH and a proton. So there are 6 categories by which enzymes are catalyzed, and they're classified by the type of reaction that they are responsible for catalyzing. The first is oxoreductases. So these are all going to end in ASE, but the first part of the name is going to tell us something about the type of reaction that that enzyme catalyzes. So in this case, this class catalyzes oxidation reactions. And we just looked at, on the previous slide, an example of that. But here we have the reverse of the reaction from the previous slide. Here we have pyruvate and NADH still in the presence of lactate dehydrogenase, in which we are reducing pyruvate back to lactate, and NADH gets oxidized to NAD+. So when a substrate is oxidized or reduced, a coenzyme is required, and that's going to serve as an oxidizing or reducing agent. So in this case, NADH is a reducing agent because it gets oxidized, and in the prior reaction, NAD+ was the oxidizing agent. Transferases transfer a group from one molecule to another. In this example, alanine transaminase. So transaminase tells me that this enzyme is going to transfer an amine group. And that's exactly what it does. Here's alanine, and here is its amino group. And it transfers that amino group to alpha-ketoglutarate. So that amino group takes the place of the carbonyl group that was present here on this carbon. And we have the carbonyl now group on this carbon, and we now have pyruvate and glutamate. Hydrolysis catalyze hydrolysis reactions. So we're going to cleave bonds with water. You've actually already seen a hydrolysis reaction, but we didn't, at least I don't recall that we indicated that it required an enzyme. So here we have a triacylglycerol, glycerol backbone, 3 fatty acids, and if we have 3 molecules of water, we will end up with 1 glycerol and 3 fatty acids as a result of that hydrolysis that is catalyzed by lipase. Isomerases catalyze the conversion of one isomer to another. Okay, so this is a reaction that is found in the glycolysis pathway. Dihydroxyacetone phosphate is isomerized or converted by triose phosphate isomerase to glyceraldehyde 3-phosphate. And so you see that this carbonyl group is moved to this carbon, the N carbon, to this carbon, and then we have the hydroxyl group on the second carbon. And again, the name of this enzyme tells us something about what is happening here. Both of these, the reactant and product, have a phosphate group. Triose, they're 3 carbon molecules, and so triose phosphate isomerize, so it is just rearranging the atoms in that molecule. Lysases, this is going to catalyze the addition or elimination of a double bond in a molecule. That is not worded properly, I see. The addition of a double bond or the elimination of a double bond. Fumarase, this is an enzyme in the citric acid cycle that acts on fumarate. And we see that we are adding a molecule of water across the double bond. Ligases. Ligate. This is an enzyme that catalyzes bond formation. So there's also energy release from a hydrolysis reaction associated with these. This process is generally energetically unfavorable. So we need the energy released by hydrolysis in order to drive this reaction. So here we have, so ligases are going to ligate 2 things together or join them. This is different from a condensation reaction, which condenses 2 small molecules to make a larger one. The difference here is this, in addition to an enzyme, requires ATP, and the energy comes from the hydrolysis of this ATP molecule. So here we have pyruvate. We have carbon dioxide. We have the enzyme pyruvate carboxylase. So that actually is going to add that CO2 molecule to pyruvate. And in order for that to happen, we have to hydrolyze that ATP and release a phosphate. So now we just have ADP left, and that's the phosphate that was released, and pyruvate is converted to oxaloacetate. This is the first, what we call the sparking compound in the citric acid cycle. So it's a link between glycolysis and citric acid cycle. So this is just a table of the classes, enzyme classes that we've just talked about, the oxoreductases, transferases, hydrolases, isomerases, lysases, ligases, and their subclasses, and the type of reaction that they are responsible for. So oxoreductases are responsible for oxidation reduction, dehydrogenases, addition or removal of hydrogens, transaminases, again, transferring an amino group. We'll see kinases in glycolysis. These transfer phosphate groups. Lipases, hydrolysis of lipids. I don't know if we'll discuss proteases or nucleases specifically, but they're more specific to either proteins or nucleic acids. Dehydrases, removal of water. Decarboxylases, remover of CO2. Synthases, add a small molecule to a double bond. Remember, we saw that in the lysase. Oh no, we didn't see that one here. Excuse me. I think we saw CO2. I'm not remembering that correctly. We did see that in carboxylase form a bond formation between a substrate and CO2. That was in the last slide. So how do enzymes work? We're just going to take a brief glimpse at how they work. There are two models. One is preferred over the other, but in general, an enzyme is going to contain what is called an active site. And that is the region of that enzyme that actually binds the substrate. Okay. So here we have our enzyme, here we have our substrate, and it will bind or fit in to that active site. And the substrate must be in that active site in order for the enzyme or the reaction to occur. So once it's coupled, then we have the enzyme substrate complex. Once the reaction has been completed, the catalyst then releases products. So here we have that enzyme substrate complex. The enzyme is freed, and then we get the products released. So as I mentioned, two models have been proposed to explain the specificity of a substrate for its enzyme's active site. Specificity here means its attraction to, okay? How specific is the interaction between the substrate and the enzyme's active site? So the first model that was proposed is called the lock and key model, and this describes the active site as being a rigid cavity. And in order to react, the substrate must exactly match the shape of that active site. So this doesn't allow for very much flexibility in substrates, but it does contribute to extremely high specificity, meaning that when I mean flexibility, it limits the interaction with other substrates that might have a similar shape that to the real substrate. So it describes specificity very well. The other model is called the induced fit model, and this describes the active site as being more flexible or having a flexible substrate. So, flexible shape. So it can adjust to accept more or a variety of substrate shapes. Okay, so it could act on more than one substrate, and that's what happens with some enzymes. So if we have a more flexible active site, we could have more than one substrate that will be accepted. And when the substrates enter, then the shape of that active site adjusts to fit the shape of those substrates. Okay. So induced fit or lock and key. So active sites do change shape after substrates bind. So here we're shown the active site of this enzyme before binding. So we've got this open pocket here. We have got a glucose molecule. And once that glucose enters that active site and is bound, the enzyme closes around that substrate for a snugger fit. So that allows different amino acids in that enzyme to interact with the atoms in that glucose molecule. So enzyme activity can be affected by a number of things. Temperature and pH definitely affect enzyme activity. There's typically an optimal temperature and pH for optimal enzyme activity. They don't all have the same optimal temp and pH. That varies depending upon what that enzyme is and where it is active in our bodies. Typically, an increase in temperature is going to increase the rate of most chemical reactions. And enzyme catalyzed reactions can also exhibit an increase in the rate with increasing temperature until that maximum activity occurs. And that's usually around 37 degrees Celsius or body temperature. So as far as enzymes in our bodies, body temperature is going to be the optimal temperature, but pH is going to change depending upon what that enzyme's activity is. And each enzyme does have an optimal pH at which it catalyzes at a maximum rate. So if you think about it, the enzymes in our stomach acid have to be optimal at a pH of around 2, whereas when we have enzymes that are catalyzing reactions in cells or cellular regions, like glycolysis and citric acid cycle, those are going to be occurring at body temperature and physiological pH. So enzymes can be controlled, and they are controlled. We just wouldn't want them catalyzing enzymes left and right. We have to be able to control them. So allosteric control is the binding of a regulator molecule to a site on an enzyme that affects the enzyme's ability to bind a substrate at its active site. It could be positive or negative control. Negative allosteric control occurs when the binding of that regulator influences the active site and makes it less able to bind the substrate. And thus, a negative allosteric regulator is going to decrease the rate of the reaction, whereas a positive allosteric regulator will increase the rate of that reaction. So this is a diagram of those two types of control, negative allosteric control, where we have our enzyme, we have our active site. Here's our negative regulator that binds on in this little pocket. And when it does so, you see that this hinge kind of folds in, so the active site is no longer accessible to the actual substrate, so no reaction occurs. Our positive allosteric control regulator, when it binds, this opens up, so it can accept its substrate molecule, and thus the reaction occurs. So we can have other substances that bind to enzymes and alter or destroy the enzyme's activity, and these are called inhibitors. If we have a reversible inhibitor, when it binds to an enzyme, it inhibits that enzyme's activity, but it can leave or disconnect itself from the enzyme, and once that inhibitor is released, the enzyme activity is restored. Okay, so this is reversible. Irreversible inhibitors covalently bind to that enzyme and permanently destroy the activity of that enzyme. So there's no reversibility here. Once an irreversible inhibitor binds, that enzyme is out of commission. And penicillin is an example of an irreversible inhibitor. And it binds to the enzyme that's required for the synthesis of bacterial cell walls. So that's how that antibiotic penicillin works, or that's the mechanism that allows it to prevent those bacteria from multiplying, because it just they can't synthesize bacterial cell walls. And here we show the interaction. We have our penicillin G, we have our enzyme with a hydroxyl group here, and we get, or that, the enzyme in the bacteria reacting with the penicillin. And once that bound, that enzyme is inactivated permanently. Penicillin has no effect on our cells because they're surrounded by a plasma or cell membrane, not the different than the bacterial cell wall. And that wraps up how enzymes work.